Hello, my name is Richard Price. I'm a professor of biomedical engineering at the University of Virginia. And the title of my talk is Immunomodulation of Intracranial Melanoma in Response to Blood Tumor Barrier Opening with Focused Ultrasound. So this study is motivated by two questions. First of all, we asked whether blood tumor barrier opening with focused ultrasound alters the immune landscape. So, we know that under certain conditions, focused ultrasound mediated blood-brain barrier opening can elicit signatures of a sterile inflammation response. But whether such a response occurs in metastatic brain tumors is currently unknown. Second, we asked whether, if the immune landscape is indeed modulated, does this offer opportunities to synergize with existing immunotherapies? So the methods for our study are shown here. So using clinically relevant focused ultrasound parameters, we opened the blood tumor barrier in intracranial B16 F1C ova melanoma tumors. We harvested the tumors and then we performed bulk RNA sequencing at both 6 and 24 hours after blood tumor barrier opening. From the transcriptional data, we then formulated hypotheses for how the immune landscape might be modified, and then we tested them using flow cytometry for immune cell representation and function in the tumor, the meninges, and in the draining lymph nodes as well. So broadly speaking, the volcano plots from the RNA sequencing data show that blood tumor barrier opening with focused ultrasound markedly augments differential gene expression in 6 hours, but this actually becomes fairly quiescent by 24 hours. Looking at the transcripts in more detail, we noted upregulation of many pro-inflammatory cytokines and chemokines at 6 hours. And since many of these could drive dendritic cell maturation, we assayed for CD86 expression, which is a marker of dendritic cell maturation on the dendritic cells in our 3 tissue compartments. So here we saw no changes in the tumor or the draining lymph nodes, but there was a modest increase in CD86 expression magnitude on dendritic cells in the meninges. We also observed the upregulation of many transcripts and gene sets that were associated with MHC class I antigen presentation and processing. So we therefore examined antigen processing by dendritic cells using a ZS green ova tumor model that allowed us to track the ova antigen. So here we saw a slight but significant change in dendritic cell antigen processing in the tumor, but there was no difference in the other tissues that we studied. To examine antigen presentation, we stained for the synfecal peptide MHC class I complex on dendritic cells, the presence of which would indicate cross-presentation of this ovapeptide antigen via MHC1. So here we saw no changes in the tumor or meningeal dendritic cells, but dendritic cells in the superficial draining lymph nodes exhibited a strong trend toward enhanced antigen presentation. So after noting these signatures, we tested whether blood tumor barrier opening with focused ultrasound could enhance the ability of activated T cells to home to the tumor site. So to test this, we performed an adoptive cell transfer of activated OT1 T cells. These are TCR transgenic CD8 positive T cells that are specific for the ova antigen. And we did this immediately prior to blood tumor barrier opening with focused ultrasound. 18 hours after treatment, however, we actually saw no evidence for enhanced T cell homing with focused ultrasound to either the tumor or to the meninges. So there are several possible explanations for this lack of a T cell response. For example, when we studied tumor endothelial cells at 6 hours after blood tumor barrier opening with focused ultrasound, we saw no changes in key leukocyte adhesion molecule expression. This includes E-selectin and P-selectin, as well as VCAM and ICAM. So one possible explanation is that there was simply insufficient activation of the tumor endothelium to recruit the T cells from the circulation and into the tumor. So in conclusion, opening the blood tumor barrier opening with focused ultrasound elicits the early upregulation of transcripts and gene sets associated with inflammation and immunity. Further, signs of dendritic cell maturation, antigen processing, and antigen presentation are evident in the meninges, the tumor, and the draining lymph nodes. However, these responses appear to be insufficient to drive activated T cell recruitment to brain tumors. One possible explanation could be the lack of augmentation of endothelial cell adhesion molecule expression. So while there do appear to be some opportunities here for blood tumor barrier opening to synergize with dendritic cell responses, the blood tumor barrier opening stimulus itself does not appear to be sufficient to actually drive a T cell response. And so finally, I'd like to thank the students and the faculty who collaborated on this project. They're all highlighted in blue here. And I'd also like to thank my funding sources. Thank you.